Okay, I'd just like to summarize some of the material from week 13, where we had a little bit of a chat about bioinformatics. So, if you look at the history of DNA and DNA studies and molecular biology, we've gone from an era where there were debates on what the actual hereditary material was, through to this idea of a central dogma where there was a one-way flow of information from DNA to RNA to proteins, some fairly basic ideas that have been elaborated upon. And also, we had the key events where Watson and Crick, discovered or postulated the structure of DNA, and then the discovery of a genetic code and how this DNA sequence is transcribed and translated into functional proteins. Okay. Now, since the sort of other things going on in the world, particularly computerization and high-tech applications to biology, have sort of pushed us into this sort of genomics era, where whole genomes have been sequenced. So there's megabases of DNA sequence material available. It's just phenomenal amounts of DNA sequence data is currently available, and the techniques we use to amplify and look at human or just DNA sequences generally through amplification and all these other things. So we live in a world where DNA is very easy to manipulate, and there's a lot of sequence data out there. So we now sort of live in what's referred to as the post-genomics era. Okay, we're going beyond just reading DNA sequences and sort of applying that information in lots of very important ways. Now, as a consequence of the amount of data that's publicly available, there's opportunities available to develop software programs to analyze this data. So effectively, if you take computational studies and mix it together with informatics and biological data, you then effectively have the ground rules and the seed of bioinformatics. Okay, and I'll just without going into too much detail about the bioinformatics sites that are around, I'd just like to sort of just quickly talk about bioinformatics. So I guess key events in the human story in terms of the sequencing projects is that we had two consortiums competing to sequence the human genome, and the byproduct of this was that the human genome sequence is publicly available for all to see and read. Okay, and the sequencing of the human genome was preceded by sequencing a variety of model organisms. So effectively, we have genome sequence data not just from multiple sort of individuals from the same genus, but we have sequence data available across different genus. So we can quite easily compare the sequence of a DNA replication protein from Saccharomyces cerevisiae with human, and we can quite easily compare a Drosophila transcription factor to a human transcription factor. And if studies have been done in one organism, that information can inform studies that are ongoing in another organism. Okay, and it allows the yeast community to communicate with the mouse community and the zebrafish community and the C. elegans community. So it's basically harmonized a lot of the research that's going on in very different labs, because they can now talk about the same sequences and similar proteins in different environments. So to deal with all of this data that is available, publicly available, it's too much information for human inspection alone. So it's a matter of applying computational techniques to solve problems. Okay, so if you have So if you can marry together the biology and the computing into a cohesive framework, then there's great opportunity to develop tools for that are useful for interpreting this data. So computers and sequence databases have been evolving for quite a while now. A typical scenario is that we have a database, which is a collection of sequence data or microarray expression data or protein structural data or phenotype expression data. There's or even just simple PubMed kind of data about publications. So the data needs to be stored in an accessible format, such as a MySQL database, a relational database. Now, within this database, there are various algorithms which can query the data, push it into a web server, and then out to a browser, so that wherever you are in a research lab, in America or Europe or Africa or wherever you are, you can access these databases remotely and process these data. Now, typically, bioinformatics for a few years was thought to be DNA sequence inquiry algorithms. So in this instance here, we're looking at a BLAST search to search, for instance, a DNA sequence from a database and push that data into a web browser. But there's a lot more that can be done with bioinformatics than simple DNA sequence analysis, although tools that can do this are very important. So effectively, if you can think of an algorithm to process data, then you can implement that within this framework of stored data being accessed in web pages. It's very easy for humans to look at genome sequence and do alignments in your head of how these align. It's a bit more tricky to write algorithms to do this for computers. But a lot of people are applying algorithms, Bayesian algorithms and other algorithms and machine learning and all these kinds of things to make more complex alignments possible and accurate over many hundreds of thousands of base pairs. So these algorithms on NCBI websites and the many other websites that exist are very good at storing all this data, and people have applied algorithms to look for raw genome sequence data and find gene sequences in there, and to annotate these gene sequences and to make this sequence data more informative to the end user. So some of the way these algorithms work is they have to align sequences that are very disparate. Then effectively what they do is they score how well two sequences align, and they do this by breaking up the sequences into small words, if you like, small fragments of sequence, and look for the occurrence of these sequences again and again in the parents in the query sequence, and then depending on how many times these words align with the data, it scores the data, and then it's a matter of matching these scores and looking for sequences that have high scores. So and effectively using this idea, you can align different sequences together. This is an example here where algorithms have been used to align a whole range of sequences upstream of a start codon, and then you can see that upstream of the start codon there are conserved regions which or motifs which are very important for the function of that DNA. So by aligning multiple sequences, you can see what's common between sequences. So these are the sequences of DNA that have not varied evolutionary over the evolutionary time frame. So you go back to yeast, you go to fly, you go to human, these gene sequences still exist, meaning that if you mutate this gene, if you evolve this DNA sequence here, then you lose function, and therefore the genes aren't expressed, and therefore there's strong selective pressure to maintain these sequences, whereas these sequences here may be less important and less functional. So if these sequences change over time, it doesn't really have a strong consequence in terms of gene expression. So by doing sequence alignments, you can see which regions, which motifs are really important to have conserved, and therefore conservation is thought to relate to important sequences, functional sequences. So effectively, using DNA sequence alignments and things, people have made attempts to interpret raw data into reading frames, stop codons, start codons, genes, promoters, introns, and all the other things that exist in DNA sequences. Now, just to be fairly indulgent, if you like, but also to give you a real-world example of how we can apply bioinformatics to real-world problems, I'm just showing you a website that that I developed over the Christmas break a couple of years ago, and this is one that's based on a MySQL database. It's got a web front end, and you can query the database to pull out information from the database, and you can add your own DNA sequence, and you can sort of query DNA sequences. But in this web page that I was playing about with and developing, rather than visualizing the sequence as alignments and what we're doing, we were converting the sequence into an audio stream so that you could listen for changes in audio patterns that relate to motifs and that relate to start codons, and that can be used to distinguish sort of a gene from a non-gene, so a coding region from a piece of DNA that might have another function. And so that's just a way you can sort of apply these processes. So effectively, what is happening now, if you look in the bioinformatics journals that are around, they're full of these kinds of tools, these sort of niche tools that address small problems. And there's hundreds and hundreds of databases available now to look at very small aspects of biology in a sort of intricate kind of way. So it's not just about clusteral alignments and blast searches, but it's about applying computers to process database information. And you can create your own database, create your own algorithm, and create your own web pages to process this data. So it's an exciting time in a lot of ways as a bioinformatician these days. So to move from a biologist to a bioinformatician, you need to have an understanding of how computers work and how code works and how databases can be built. And it's not difficult information, but it's not information that it's kind of cross-institutional information. It's computational information within a biological domain. So by using computational languages and databases, we can create workflows, and then we can sort of use these to create tools. Another tool which I've built, but is that there are lots of other tools that use this very same analysis. So it's not as if I've invented the tool, I've just applied the tool to my problem. And this kind of bioinformatic analysis is simply comparing two gene sets. Okay, now we could have a database of thousands of gene sets, and we could have a few gene sets of our own. So, and then we compare our data, such as set A, with the database sets, which is set B, and then we look for an intersection of those two sets that satisfies some statistical analysis. So if I take my gene set of interest and I compare my gene set against a couple of thousand gene sets that exist in a database, and then I process them, I can work out which of the database sets are close to my sets. And that way I can sort of start to say, well, my genes, my experiment was similar to another person's experimental results, and then I can look at that paper, track that paper down, and start to really sort of compare and think more deeply about the data. So again, that would be another example of a niche application. So by using the database, a bit of coding, and the web front end, we can design these web pages. And the software that's used for this is open source, it's free, and it's available to anyone who cares to download it, which is actually really good. So XAMPP, will give you database tools, some coding tools, and some web front end tools, all in one package. And then you can set up your database, find your data, import your data into your database, and then start to build your database. And then once your databases have been built, you can then set up a front end to query the data. So for instance, I've built a database from 130 papers, 640 gene sets, and then out of those sets of data that exist in the database, I can then take other people's raw data that they've just generated in a lab, and I can intersect it against the database and find which other experiments in the literature have a similar result to the new set that someone's just generated. And that is a really good way of relating your data to the published literature. So, so this is just applying, a database, the yeast deletion library database, where you've got my experimental results against the database results, and I look for a common intersection, and I consider all genes in the genome when doing this analysis and calculating this statistical significance of this intersection. I read a quote somewhere that the only certainty in computer programming is that every time you modify your code, you introduce a new bug, and I thought that was quite telling, because I'm forever introducing bugs into my code. This is just a header from a web page I designed a few years ago, and just by way of encouragement for you guys to think about these problems, perfect is the enemy of good. So often a bioinformatic tool that does a good job and that is available and functional is much more important than some idea of perfection that people are pursuing but never finishing. And and a lot of these highly cited bioinformatics tools that people are generating all over the place, they're simple tools that do a reasonably good job at what they were designed to do. Okay. And this is just showing another web page that I've designed, and looking at the yeast gene deletion library. And again, like I said, I've collected the data from papers. We run intersect tools, doing this statistical analysis of what's common between the sets, and then we go through and have lots of tools which are built in there for the analysis of the data. So that's my sort of take on bioinformatics. It's not just related to gene sequence analysis, but it's a way of processing the large volumes of data that exist from many experimental protocols and designs across many different model organisms. So find your niche, work out how you want to interpret the data, and then script it and make it available as a method, as an algorithm that other people can use. Okay, thank you.